could also reprogram. And so this was just published in 2002. Now, an exciting development, just yesterday, there was a press release of the first human embryonic stem cell line that was made through cloning. So I already told you about Thompson's work in making human embryonic stem cells by taking embryonic stem cells out of a blastocyst and growing them in culture. This is a variation of that where these scientists, they took cells from a baby. So this is an 8-month-old baby who was born with a very rare genetic disease. They took nuclei from these skin cells, and they basically injected them into a human blastocyst, which was, again, now an egg that had been discarded by a fertility clinic. The egg nucleus was removed, and this child's skin cell nucleus was inserted. So this is exactly how Dolly was made. So in theory, this is exactly how you could clone a human, except the scientists stopped at the stage of the blastocyst, and they went in and they took out those embryonic stem cells which had the DNA from this patient. And they now have a cell line growing in the lab, just like those embryonic cell lines that I told you about, but that were generated through this kind of nuclear transfer. So this is something where one could argue, and we can maybe talk about it later, that if they had instead taken that embryo and implanted it, they would have generated a cloned human, and it might have worked. I'm not going to comment on whether it will or should or whatever. So this is a great cover from Time, and this is actually, I've had this probably for 20 years. I don't know what year this is, but it says, human cloning is closer than you think. And I think this experiment